The overall goal of this procedure is to conduct measurements of the phosphorus release from lake sediments for estimation of the internal phosphorus load. This is accomplished by first collecting intact sediment cores in the field. In the second step, the sediment cores are exposed to redox treatments and incubated in an environmental growth chamber. Next, the water overlying the sediment cores is sampled for phosphorus concentrations at regular intervals throughout the incubation period. Finally, the phosphorus release rates are calculated and the internal phosphorus load can be estimated based on the changes in phosphorus concentration over time. Ultimately, the laboratory-based sediment incubations are used to quantify internal phosphorus loading to lakes, identifying the role of the sediments in lake eutrophication.
The main advantage of this technique over other methods for estimating internal phosphorus load, such as phosphorus mass balance and in situ changes in phosphorus concentrations over time, is that this method can be adjusted to answer a variety of lake management and research questions.
For example, with this method, the relative importance of internal versus external loads to the lake can be analyzed to inform lake management in eutrophic lakes, as well as effectiveness of treatment efforts to reduce internal loading.
Conduct the water quality sampling first. At a minimum, measure water depth and vertical profiles of water temperature and dissolved oxygen. Collect any other water quality data and samples that are desired to fulfill the specific goals of the study at this time as well. At each sampling location, use a Niskin bottle to fill